So I got the feeling from the introductions that pretty much everyone in here is involved in casework. Am I correct? So everyone has done differentials. Yes. Okay. So I'm not going to go too lengthy in this because I'm assuming you all know this, but we thought we'd just give a quick 10-minute talk or so on your basic cell identification. Oh, I'm sorry. Yes. Because they're out of order, like she mentioned, you want to go ahead to your basic cell identification PowerPoint in your paperwork, and I'll wait until you think you're all ready. I want to say it was more towards the front.
It's like third from the back, I think.
Oh, is it? Okay, so what I have here on the projector is the mixture of spermatozoa and epithelia. This is what you would typically see using Christmas tree staining. If you took a slide, let's say, out of your sexual assault kit, or you made a slide during a differential. This is using the 20X objective. Hopefully my boss is proud that I said that correctly. And as you can see here, what I did was I circled some of the spermatozoa here. They're very small in this image. But this is what most of you typically see when you're doing a differential and you do your Christmas tree staining. And the red isn't showing up very well on here, but you get the gist of it. The nuclear material is stained red. I pulled this out of a histology book. This is a basic spermatozoon. I don't know if I pronounced that right, but of course, for our purposes, mainly what we're interested in is the general structure of the sperm cell, the head, the neck, and the tail. A lot of times with what we're doing, most of the time, we don't even see the tail. So that's something that's fairly important for us as the forensic biologist in trying to look on our Christmas tree stain slides. We really are having to look for that head and identify the head and make sure we've got a sperm before we're going to, let's say, do laser microdissection. Of course, we're not so interested in the, let's see if I can use this fancy thing here. We're not so interested in the more detailed things here. Of course, it would be nice if our sperm had the name written on the head or on the tail for us, but we're not so lucky. But generally, we're interested in the head, the neck, and the tail. And of course, the head especially, for our purposes, generally when there's Christmas tree staining, it's staining the nuclear material. So we're getting this area here. And then, let's see. The nuclear material is stained red. The acrosome, which is this top portion, is clear. So those two things, at least for me, help me to really be able to feel more confident when I'm looking at sperm on a slide, especially if it's just a head and no tail. And what I generally do is I'll take that fine focus and kind of move it in and out, and that helps me to really see that acrosome versus the red stained nuclear material. Okay, so this is what we saw the first slide, our Christmas tree staining 20X objective. Oh, what happened there? I don't know what happened. Okay, next is just some epithelial, dissociated squamous epithelial cells. This was taken from the Atlas of Human Histology. And this was just to, help you all visualize. Of course, this is what our epithelial cells generally look like. They're not in the fresh state when we get them. They're dissociated. So, or actually, I don't know if that's the proper term, but. Okay, so here's a 40X objective of the same slide that I just showed you. The increased magnification and resolution that you get with the higher objective, you start to see the epithelial cells when they're not in the state that we just saw. They take on many forms on the slide. So for example, in this one here, you see it's folded. Same here. But sometimes you'll get these nice looking ones like this one. And of course, with laser microdissection, we're especially interested in the nucleus. So the nice thing about Christmas tree staining, and even though this picture isn't very good, that nucleus generally will be stained red, and so that helps us to identify that nucleus and cut it out. Okay. And this is at 63x objective, and now it's actually nicer. You can see the red a little better on the slide, and that is, 63x objective is much better to see. And what's nice about the laser microscopes, I'm not sure about all the other systems, I'm assuming they're probably the same. You have a screen where you can see a nice big image, and that's one of the great advantages of this technology. Okay, now, I put these next images in here because this is an epithelial sperm cell mixture on the pen slide, the polyethylene naphthalene slides that you need to have when you're using a Leica microscope. And I think Pat mentioned this. As you can see here, first of all, this is using the 40x objective. And as you see, there are lots of these tiny, tiny pores with these slides. Now, that's something that can be seen sort of as a disadvantage, because you see here's a sperm cell head with a tail, and it's not much bigger than these pores. So that was one thing at first when we were using these slides and using the Leica microscope, we were having sort of, it could be hard, especially when looking at using a lower objective to really identify those sperm. But another thing I want to point out about the slide, this is not a Christmas tree stained slide. And Christine's going to go into this more when she gives her research presentation. She did a lot of staining studies that I did not do. I actually took from her, and some of that had to do, again, with the DNA typing downstream. But what I do with these slides is I just do a quick, like 2 to 3 minute staining with the nuclear fast red, and that's it. I rinse it off and go. Because that way, with the green, first of all, the green, she thinks that it inhibits PCR. Because it's an acid. And then also what's nice about this, as you can see here.
Sorry, what was the staining called? Pen?
Nuclear fast. Oh, the slide itself, it's called a polyethylene naphthalene slide. And don't get me to spell naphthalene. But that's the type of slide that Leica, that you have to buy in order to use a Leica microscope. It's a glass slide, but then it has this film that's glued around the edges to the slide, and it's actually, you prepare your sample on this film, and then it's placed upside down on the microscope. The laser goes through the glass. It cuts the film, and it's the film with the cell that drops by gravity.
Okay.
And it's the film that makes these pores.
Okay.
That's got these pores on it. So you can see it's pretty grainy. It's a little bit, getting used to, but.
And what staining did you say there is none, or that you use?
This is nuclear fast red staining
okay.
only. This is with your Christmas tree stain, but it's just the red.
Right.
So you're only using a chronic shot?
I'm sorry?
Oh, we call it chronic shot, nuclear fast red?
Yeah, that's just the red.
So you're not using the picaron to go carbon?
No green. No green.
Okay.
This again was Christine. Hopefully, we'll go into this a little bit with her presentation tomorrow. She did a lot of staining studies, not just with Christmas tree staining, but with H&E stain, and she did fluorescent, staining. She did all kinds of staining, and because Christine had bought this microscope before we did, and then we bought it, I of course called her quite a bit, and she helped me when she could, and this was something I picked up from her. Was just because again she thought that the green may inhibit the green staining because it's acidic, that it may be causing some kind of inhibition with the downstream PCR analysis. So that's why I went with this, because we had it in house already.
Yeah. How long do you stain with the?
With this, it's only 2 to 3
right.
minutes that I
That's
put the
it.
stain on. I can't remember what Christine does. Yeah.
What did you do normally for like a differential? Did you only stain for 2 to 3 minutes normally?
No, we did like 20
15,
minutes.
because we use
Yeah.
15 for the chronic shot, so that would be reduced.
Yeah, so which is nice. It's much
Yeah, you
quicker.
only need 2 to 3 minutes stain.
But still, look, you can see that it's picking up the red in just even the 2 minutes that you're staining. So anyway, that's what this depiction is. And then I then focused in same slide, took a picture of it at 63x objective, and you can see this is nice. I mean, this is a nice image. You see it on the computer, and you're able to quickly pick out, your sperm, nuclear fast red, in the nuclei of the epithelial cells. Going to your question about thickness, you know, you could see something like this sperm here. You know, it's right next to a epithelial cell nuclei. And I probably wouldn't cut something like that. Instead, I would try to cut something like this. Okay, so here I have a labeled picture of our mixture of spermatozoa and epithelial cells with the pen stain, or the pen slide nuclear fast red stain only. And just to point out, again, 63X objective. You're gonna, when you make your cut, at least with the Leica, you're gonna make a nice circle around your red nuclei and cut it. And then, of course, with your sperm, this is a beautiful, of course, beautiful sperm. But we all know with forensic evidence, we rarely see, I mean, it's rare if I see something like this. So this is a sperm with a head, neck, and tail. And you notice here the nice cap, you know, the acrosome, that clear acrosome on top, the pink nuclear stained sperm head, and then, of course, your beautiful tail. But again, we don't see that very often. But here, you know, you can see that's a sperm head. And that'd be something that would take a little more skill. You would just want to make sure that that's a sperm head that you're cutting and not a pore on the slide or something. Okay, so again, I'm talking about, I might be hitting you over the head with this, but the acrosome is the important, to me, the important feature of being able to identify your sperm head without its tail. You know, lots of times when you look at these differential slides, there might be yeast on the slide, you know, there might be red blood cells, just a pink blob, you don't know what it is. So again, you want to make sure that if you're cutting, because this is what we're cutting now to help separate our epithelial cells from our sperm cells, I do a fine focus and I really try to make sure that that head has that clear acrosome on top. And that's it. Any questions about the basic cell identification? Okay. I think Christine is our next speaker.
What I'm going to do is, hopefully this won't mess up the AV guys. I'm going to actually change the curriculum a little bit because I see a gap in information before we get into the lab. So if you look at your schedule, right now I'm supposed to do lab practical overview before we go into the lab. But I'm actually going to give you a little more information. If you see, after lunch I'm supposed to do LMD dissection of sperm cells from a mixture. I'm going to give that right now. And then I'm going to go over the lab practical overview. So the PowerPoint should be, let's see, I'm going to pull it up. Should be this PowerPoint, LMD of sperm from cell mixtures. I'll wait till you guys find that.
It's up in the beginning.
And unfortunately, the screen doesn't seem to display the color as it does on my computer screen. Because this stain, I mean, it looks like this is the Kelly's, but this is actually a Christmas tree stain. And then on my computer screen, I could see the color, but in there it looks pretty black and white. So hopefully you'll actually still see the slides when you're looking at the instrument. You'll be able to see the different colors. Okay, so why are we here? And you know, we're talking about cell separation. And just to review, you know, DNA mixtures are frequently encountered in forensic evidence, particularly in sexual assault crimes. And the idea is that we want to identify the individuals involved in the crime. So genotype mixture interpretation and then the statistical analysis can be challenging and time-consuming when we have DNA mixtures in a sample. So the separation of the different cell types can provide cell fractions from individuals involved in a crime. So So we want to do separation. And just a review, there's some different separation methods. The most common and the gold standard used right now is a differential extraction. And Pat will go into that a little more detail later. Just some other different methods. There's flow cytometry, microchip separation, membrane filtration, magnetic antibodies. And these, there's some of them have been successful, some of them not so successful. Actually, I don't know how to put it in here. There's a lot of work being done on microchip separation, and I'm kind of excited to see where that goes. But there's also, I wrote down here, Y chromosome. Even though it's not really a physical separation method, it is obviously used in DNA mixtures and extremely useful, although it doesn't provide the statistical power that your code is 13 can provide when you have a mixture. So we propose, of course, that we use laser microdissection to separate your sperm cells from your epithelial cells. And just here's kind of just a real simple diagram. Actually, I'll write on here. So on the top, you'll see will be your microscope objective right here. And then the laser beam in red that you see, that's where the actual laser beam comes through the microscope objective. And down here is the slide. And you'll see that what you do is you have this slide, and I have, I guess, a layer. This is an old version. There's actually a layer. Let me get another highlighter here. There's actually a layer here, if you can see it there. That would be the pen film that's already prepared on the slide. And then you prepare your specimen smear on that pen film. And here you can see those are just examples of your cell, a couple cells on the smear. You'll notice that the slide is actually upside down. That's how you put it on there after it's prepared. See if I can. And this is just a diagram of the laser. A blade goes through the actual slide and cuts around the pen film. And then that sample with the cells and that pen film drops into the tube. So that's just the basics for the Leica microscope. Again, here's that same. This is what you'd see under the microscope on the computer screen. You've got sperm cells here, and obviously you can see the big epithelial cells. So there's a couple technical issues of sperm ID. Kelly mentioned them, some of them, using LMD, especially under just bright field conditions. She talked about the pores in the pen film. So you see all these little, these little pores, and it's kind of this architecture that's in the film. And that's actually the biggest obstacle. There are some newer slides that are coming out that we're hoping, and we have some actually here that were just started being available maybe in May that we're going to test that are much clearer. And in addition, and I don't know if Leica Microsystems has been working on this, oddly enough, those pores have been purposely put in there because the original manufacturing of the pen film wasn't designed for laser microdissection. It was designed, I believe, for like audio video technology, which they wanted the pores put in. So there hasn't been that evolution of making this product exclusively for laser microdissection. The reason why they hadn't fixed it is originally these slides are for cryosection tissues. So when you have a huge tissue section, you don't see those pores. But now we're dealing with smears, so the pores make it more difficult kind of to search for your sperm sometimes. So just some technical issues. Hopefully, again, these things will improve as they have more products and different and clearer slides. So again, the pen film architecture. So that's why I say right now, really, a stain is necessary because if you look at an unstained slide, it is actually kind of hard to see unstained sperm with all those pores. And when Pat talked about using DIC, those pores even get worse if you use DIC at this point. One of the things that you can do when you're actually working on the microscope and trying to see, is that a pore or is that a sperm head, is you can adjust the focal plane to kind of identify your sperm, and you probably do that also, just normally in your sperm ID. And again, I said there's these new slides we're going to try out. They're very clear EP director slides. And what do you do? What about high density of epithelial cells that was mentioned? Here's some possibilities. You may need to dilute your pellet out and smear it over a larger area. In addition, there's no cover slip on these slides. So again, it's not as clear as you might expect when you're doing your normal sperm ID. And also you're looking through, remember that slide I was saying or showed earlier, your slide is upside down, so you're actually looking through the glass slide at your sample. She just says the technical issues, and really it takes just more practice, just looking at lots of slides, and after a while you get used to these little idiosyncrasies of the specimens. I just put this set correction collar objective, just something about, since you're looking through the slide, you do have, and it'll be adjusted for you in the class, but it's something that if your correction collar isn't adjusted for the 1 millimeter, that can really mess up your image. So it's something, if you don't get a clear image, just kind of think you might need to set your correction collar on your objective to 1 millimeter. So this is kind of the basic look of the computer screen. Now this is an older version of the software. You will have a newer instrument with the newer version of the software, but the basics are the same. Well, you have a control panel, and you'll see your image in the middle. And in this image, the analyst has outlined, circled the sperm cells for dissection. And then we will activate the laser, and at the end, you'll see holes where you cut your sample. And then on the right here, it says sperm cuttings in tube. You can actually look into the tube and see your cuttings in there. If you could really see, if you look close, you could actually see little sperm heads. The epithelial cells are obviously easier to see. And you can also collect epithelial cells if you want. This particular cell, if you could see, it's actually the same shape if you flip it as that cell. So especially if you're doing low copy number work, when you do a lot of cells, you just see a cluster of cells, you're happy that you've collected it. But if you do low copy number work and you only have 5, 10 cells, you can actually look in the tube and say, yeah, I'm confident I see them in the tube, they were collected. This is just to show the current software just has more control panels. We'll go in and see actually and work on the instruments. But this is just kind of the look of the new software, which is actually very nice. And I'm going to run a video, and I got to go search for it. Oops. Oops. Now this is, oh, it's actually already running. This is a homemade video, and I'm sorry if it's hard to see. You'll see it in action in there in the room. This is actually the microscope objective right here. Oops. Let's see. It doesn't come out well with the dark. But there's microscope objective. Oh yeah, there we go. And then you'll see that there's the slide on the stage. You'll see those circles. We're going to be putting those circles on there. That was just so I knew where my spot was. And the laser, it comes down through the objective and actually cuts through here. In the video itself, I think I stopped the video or paused it for a moment. You'll actually see a blue light cutting the sample. Let me see if I can get this running again. Go back to the mouse. See that it stopped. Okay, we're back going. So in my homemade video, once again, here's an example of, if you actually want to collect the whole cell body of an epithelial cell, you can. Somehow it skipped over the sperm one. We can go back. And this is about the speed of the old instrument. And this is in real time. The new instrument you will be working on is actually much faster, which is great. And you can see it's cutting through. So it's going through the slide, cutting around the membrane, and you'll see the sample drop. And that it's going into the tube. And in this next example that it's cutting, I'm just cutting the the nuclear material. Now we will be being focused on cutting sperm, but you'll have the opportunity if you want to cut epithelial cells. I don't know why it's skipped over a certain part. Let me just try to find it. Okay, yeah, this is just the sperm. Somehow it skipped over this part, or I didn't see it. This was just the sperm cells being cut out. Again, it'll actually be faster in the lab. I was running in about 20 minutes I could cut 100 sperm cells. And we'll see how fast you guys can cut. The laser now is more powerful, and so you can cut faster, which is great.
How many sperm cells do you think you need for a full profile?
Well, we'll talk about that a lot tomorrow on the research. If you used to use standard, just briefly, you use a standard PCR conditions, like what's mostly validated in your forensic laboratories, I'd say 100 cells is really the bare minimum. But I did a lot of work on low copy number work, so I went down to five cells successfully to get profiles. Yeah, and we'll talk about a great deal tomorrow about that. Okay, so that was I just wanted to give you that background because we kind of had this gap of even showing, okay, what the Leica Microsystems instrument does. Now we'll just switch over to our normal schedule and talk about the lab before we go in there. So I don't have a PowerPoint. We're just going to look at one of the pages, and I'll find it in here. It's probably just a few pages in. I'll hold it up so you know what to look at. It's this one that's colored, and it's a lab practical flow chart. So basically what's happened before you even got here is that the staff back there has been working hard, getting everything together, and they've been preparing sperm and epithelial cell mixtures. So I provided them with neat semen samples, and they took some buccal swabs from their employees, and so I had them extract and make samples from this. And basically we have, for every buccal swab, we have about 5 microliters of semen. And we kind of took a really brief look at the samples, and some of them have a lot of epithelial cells and some have very little, but that's kind of typical of what you might see in regular casework. So there's a variety there, and it'll probably be just kind of random which tube you happen to pick. And what you will be doing is preparing the slides for LMD from those mixtures. So you're going to go ahead and take those mixtures, smear them on slides. I'll go through that when we're in the lab, and stain those slides. You'll also in there, we'll give you a demonstration of the instrument. You'll first have Andy Lee will kind of giving you what he calls care and feeding of the instrument really briefly, just so you know how it works a little bit. And then we will go in more detail. Both Kelly and I will sit down, kind of split the groups in half, and sit down with you and talk to you more about actually how to cut it and how the software works, because that's what you'll be doing. Then you will cut particular samples. We'll have the different groups collect different types of samples from the slides. And we'll be there the whole time to kind of help you to do your cutting. And what we have to do is, because we have 2 instruments, we have 12 people, we're going to split the group in half. While half of you are doing your cutting, the other half will be preparing yet more slides for tomorrow. And these are going to be fluorescent slides. So we're going to do it all in the same room. We'll see how it all works. But you're going to be prepping some slides. And that group that's prepping the fluorescent slides, you kind of have a lot of downtime, because we're giving basically an hour for everyone to cut their samples. And with the staining of the fluorescent slides, you'll probably like add a reagent, walk away for a while. So you have a lot of break time in there. But I think it's good that you're all in the same room. You can kind of watch what's going on. And then, so you cut all your samples, and at that point we'll be getting close to the end of the day. And you'll basically hand those samples off, and the staff will finish the extraction. They're going to be doing the PCR overnight, and then tomorrow morning they're going to put it on the CE. We're doing the Profiler Plus for these samples. And I wrote down here, they're going to be 50 microliter reactions, and we're doing at a low copy number level, so 34 cycles. So something to be aware of. You need to wear your gloves. I think I did ask them if you feel like you want to, you can wear a mask. I recommend that if you do set this up in your lab and you do low copy number work, and you're going to have to set your own protocols for being really careful with that. We're going to be just a little more flexible here, and hopefully we won't have any contamination issues if you don't wear a mask and things like that. But make sure you do wear gloves. And okay, so you can see also, we'll just go over day 2. They're going to be running those samples on the CE while we continue our lectures. And then you'll be back in the lab to see the fluorescence lines that you've prepared today. We'll be running some new software and some advanced operations. You'll be able to maybe get on the instrument a little bit and cut, but we're not actually analyzing these samples. And then toward the end of the day, you'll actually see your data, and hopefully it'll all work, knock on wood, and go over that data. So what we're going to do now then is we're going to go into the laboratory, and the first thing we'll do is, you can sit anywhere. I'm going to put you in groups, but for right now, you could just sit anywhere, and we're going to start preparing our slides. And then while we wait for those to dry, Andy will go over some of the instrument components.